Hello, my name is Siti Jasmina binti Sheikh Husainuddin, metric number 180880. I'm going to talk about enzymes. So basically, the function of an enzyme is to speed up chemical reactions. It's a catalyst, and there are several types of enzymes, and each type of enzyme has a specific substrate that it will attach to catalyze the reaction, and it will then detach from the substrate, it'll detach and it'll forms products. The shape and the size of the enzyme is very specific. The shape and also its chemical properties, they are very specific, as if the enzyme is denatured and if the shape is altered, the enzyme cannot function anymore. It's denatured. The active sites of enzymes are very important, as it's the place where the substrates get attached to. As I've said earlier, enzymes speed up reactions, and if there was no enzymes, a lot of chemical processes in the body would happen very, very slowly. So basically, enzymes they lower the activation energy of a reaction, and that's how they make the chemical reactions faster. And enzymes are not consumed during a reaction. They are not produced during a reaction. They are just used. They just catalyze a reaction. Enzymes also stabilize the transition state of a reaction. Enzymes can be denatured by several factors such as pH and temperature. If most enzymes function at a body temperature about 37 degrees, and if the temperature is too high, the enzymes will be denatured. But the rate of enzyme activity can be increased with increased temperatures, but if it's too high, it will be denatured. So that's all. Thank you.
Hi, my name is Shirley Chinseli, and my matrix number is 180864. Today I'm going to tell about enzymes. Enzymes are typical proteins that act as a catalyst which speed up chemical reaction that would take far too long to occur on their own. Enzymes speed up the vast majority of the chemical reaction that occur in cells. Reactions that break down molecules, such as those involved in digestion and cellular respiration, and those that build up molecules, all require enzymes. Each type of enzyme has specific shapes that complement the structure of its substrates. The substrate is the molecule or molecules that the enzyme converts into product. The substrate fits into an indentation in a globular protein called the active site. The shapes and chemical properties of this active site are critical to the enzyme functions. Next, I'm going to tell the procedure of this experiment. First, locate the 15 ml conical tube, label stop solution, 1.5 ml substrate, enzyme and buffer. Label each of the tube with your initiates. Label 5 cuvette E1 to E5 and label it only the upper part of the cuvette face. Label the two remaining cuvettes with start and ends on the upper part of the cuvette. The cuvette will serve as control time points at the start and end of the reaction, and neither cuvette will contain enzyme. Next, using a clean DPTP, pipette 500 ml of stop solution into each of the 7 label cuvette. The stop solution is a strong base, so avoid getting it on your skin or on your clothes. The next step will be locate 2 empty 50 ml conical tubes and label one with enzyme reaction and another with control. Using a clean DPTP, pipette 2 ml of 1.5 ml substrate into the 15 ml conical tube which has labeled enzyme reaction, and use the same DPTP pipette 1 ml of 1.5 ml substrate into another conical flask which label control. The next step will be label 1 DPPTE for enzyme and another C for the control. Only use the DPTP labeled E for the enzyme reaction tube and the 1C for the control reaction tube. Okay. So after doing this experiment, our result have recorded in the table. So thank you.
Good morning, my name is Raja Aiman and my matrix number is 180878. Okay, today I will be sharing with you our experiment we did 2 weeks ago. So the aim of the experiment is to determine the rate of reaction in the presence or absence of an enzyme. So the introduction is we will be doing an experiment we conduct with the enzyme and the substrate we use, and then we'll check on the enzyme rate reaction and we'll get the results from there. Okay, so first what we do is we require 3 conical tubes whereby we have the stop solution and substrate and enzyme and buffer. So sorry, there's 4. And then we need 7 cuvettes, which is E1 to E5, and then there's a start and end. So what happens next is we have to use a DPTP, which is a disposable plastic transfer pipette, and we extract 500 microliters of stop solution and then put it into 7 cuvettes. And then we have to relabel ER, which stands for enzyme reaction and control, into 2 conical tubes, and then take 2 ml of substrate and then put it inside the enzyme reaction tube, and then 1 ml into the control tube. So what happens next is 500 microliter of buffer is inserted into the control, whereas 500 is also we put it inside the start. And then we took 1 ml of enzyme and pipette it into the ER. So we start the time, that's when the enzyme starts to take action and take place. So for every interval, like 1 minute, 2, 4, 6 and 8, you extract 500 microliters into the respective E1 to E5 cuvette. So after that, you will be taking the reading from the spectrophotometer, and there's several procedures there where you need to put the blind first to calibrate, and then only you get the results. So the results are as shown. For E1, it is 0.089, for E2 it's 0.196, E3 is 0.202, E4 is 0.186, whereas the E5 we got it at 0.170. So that would actually show the value of the absorbance, and then we have to compare with the standard which was given. So from there we can actually relate how the enzyme reacts with substrate or without the substrate. So that is all. Thank you so much.
Hi, my name is Edlinson. My matric number is 175282. Okay, last week we have done the experiment on practical 3, which is enzyme kinetics. We have determined the reaction rate in presence or absence of an enzyme, and also we measure the amount of product formed. Okay. First, I'll talk about enzymes. Enzymes are typically proteins. Some nucleic acids have also been found to be an enzyme that act as catalysts, speeding up chemical reaction that would take far too long to occur on their own. Enzymes speed up the vast majority of the chemical reaction that occur in cells. Reactions that break down molecules, such as those involved in digestion and also cellular respiration, and those that build up molecules, such as ones involved in photosynthesis and DNA replication, all require enzyme. Each type of enzyme has a specific shape that complements the structure of its substrate. Okay, many chemical reactions that enzymes speed up can occur at a much slower rate without the enzyme. Enzymes speed up reaction by positioning their substrates, adjusting their bonds so that they become unstable and reactive. Okay, in this experiment, we have studied about cellular bias. Cellular bias is involved in the last step of the process of breaking down cellulose, a molecule made up of bundled long chain of glucose that are found in plant cell walls, to glucose. This is a natural process that is used by many fungi as well as bacteria, some present in termite guts, others in stomach of ruminant, and also in compost pile. Okay, the natural substrate for the enzyme cellular bias is cellobiose. This is a disaccharide composed of 2-beta glucose molecule. However, when scientists study enzyme function, it is best that if there is an easy way to detect either the amount of substrate that is used or the amount of product that is formed. Okay, we have done to measure the amount of product formed. Okay, we can use an instrument called a spectrophotometer, which quantitatively measure amount of yellow color by shining a beam of light through the sample. The spectrophotometer measures the amount of light that is absorbed by the sample. The darker the color of yellow the sample is, the more light is absorbed, and thus the more concentrated the sample. Thank you.
Hello, my name is Yiling. Metric number is 180893. Today we will talk about how to determine the reaction rate of an enzyme. What is the reaction rate of digestion of cellulose in the presence or absence of an enzyme, that is cellobiose. In this activity, we will compare the rate of breakdown of p-nitrophenyl glucopyranoside into glucose and p-nitrophenol in the presence and absence of cellobiose. Enzymes are molecules that increase the rate of a reaction but are not used up in the reaction. Because the enzyme can keep processing the substrate over and over again, therefore very few molecules of enzymes are needed relative to the number of molecules of substrate. Because it is difficult to add really small amount, therefore our instructor has already diluted the enzyme with buffer solution, and this allowed us to easily add the enzyme with buffer solution. This will allow us to easily add the required volume that still contains very small number of molecules of enzyme. However, to ensure that the buffer in which the enzyme was diluted does not affect the rate of formation of the product, a control reaction containing just the buffer will run alongside with the reaction containing the diluted enzyme. To the first reaction tube, we'll add enzymes into a solution of substrate and determine the initial rate of reaction or product formation. In the second reaction, which is the control reaction, it will have the same buffer added to the same substrate but do not include enzymes. By this way, we will be able to compare the breakdown rate of p-nitrophenol glucopyranoside to glucose and p-nitrophenol in the presence of enzyme and the presence of control buffer. That's all from me. Thank you.